Okay, welcome everybody. Today we're really excited to have Dr. Bhowman here to talk to you about his work at NRC. He received his PhD in plant physiology and molecular biology from, I'm going to say how do you say it?
Kagawa.
Kagawa University in Japan. Following his postdoctoral fellowship in Japan, he received a Killam postdoctorate fellowship and an NCERT one, and now he's joined NRC here in Saskatoon since 2009, where he serves as a senior research officer and a member of the cell technologies team. Pankaj has an additional role as a scientific support lead for the NRC Sustainable Protein Production Program, where he works closely with small and large companies in the plant, biotech, and agri-food sector, academia, and other government departments to establish multi-organizational research collaborations with external stakeholders. So Pankaj and I worked quite a bit together over the last few years, so we really enjoy our collaboration and doing stuff together in the area of plant proteins. So he has more than 20 years of experience in the plant cell technologies and tissue culture. His research focuses on the improvement of crops through tissue culture, transformation, and gene editing. So without any further ado, thank you, Pangash, for coming to our little webinar and sharing your insights. So I'll let you take it away.
Thank you, Mike, for inviting me to share some of our recent works. So what I would like to do for today's talk, give you a very high-level overview of about NRC, who are not familiar about NRC and our program, Sustainable Protein Production Program. Then I will share some of the recent updates, that how, as Mike mentioned, that how we are deploying the cell technologies and the gene editing tools for pulse crop improvement. And as an example, I'll share my work with peas. So very high level snapshot of the National Research Council of Canada. As you know, that we are the largest federal research and development organizations in Canada, and we have total 14 research centers. So NRC Saskatoon is part of the aquatic and crop resources development research centers, and we have over 3,700 scientists, engineers, technical officers. We have also industrial research assistantship program we call client relationship experts across the country, and the annual expenditure is close to 1 billion, including the 271 million in contribution funding to small and medium enterprises. So the program, that one of NRC's program that we call Sustainable Protein Production Program, it's a 7 years program that I'm the scientific support lead for the program, and it aligns with the Protein Industry Canada Global Supercluster. Now that National Research Council is supporting that clusters, and our goal is to moving the plant protein and co-products of the value chain by supporting Protein Industry Canada initiatives, work with the academia, industry partner, and other government departments, and address the challenges through the collaborative research projects. And the 4 areas that we are highlighting or emphasizing is that food security, the environment, or the changing environment, and climate change. Of course, the economic growth, are making the protein crops production and processing more profitable. The 3 specific focus areas under our program, we call the advancing the genomics resources, because that's NRC's strength. We have worked several years on multiple crop species in terms of the genome assembly, bioinformatics. So that's our creating the resources for protein crops, focusing on protein yield, the quality traits like flavor or off flavors, and also the disease aspects, in this case, the root disease for peas and lentils. And then under sustainable production, we focus mainly on developing technologies that that can be deployed for precision agriculture. The inoculant is another good example. That's also tied for sustainable production or less fertilizer application. Being pulses being legume crops, right? And then our third focus area is the processing. How can we enhance the processing and manufacturing innovations? In fact, currently we have a call open for industry for technologies that could be deployed for plant protein processing and value addition. Okay, now moving back to my research, the team that I'm part of, we call cell technologies team. We do a lot of gene editing. So this is basically, if we categorize our expertise into 4 different, categories. So a lot of, double haploidy microspore cultures, that we have expertise that we deployed in multiple crop species. Now we are trying to deploy in peas, lentil or chickpeas, and then our transformation platform, tissue culture and transformation platform is also a long-term expertise that we have here in multiple crops using the agrobacterium or biolistic or novel method of transformation depending on the crop species we are talking. And then the gene editing or genetic engineering trait development parts, using mutagenesis, RNAi, or the latest the CRISPR-Cas tools. We learn that for trait development also the analytical skills or characterization or high throughput phenotyping, whether it's NIR or your weight chemistry, that's also critical for trait development. So we have that expertise in general. So why are we all doing this or trying to deploy the cell technologies? Try to be a crop agnostic approach that in multiple crop species we could do, because that because of the recent advances, how the transformation efficiency improved, the frequency of gene editing, several papers every month, across the world coming with the different modified enzymes or a different way of transforming or different explants. So that's all to create the variations directly in the elite germplasms, right? So we may have the variations in the existing germplasms, but the time it takes 10 to 12 years in the conventional breeding. So can we accelerate the genetic gain or variety development? That's the whole idea. And how can we save time and resources? So that's the idea of applying cell technologies and genome editing tools. This slide, just to show you that, it's a crop agnostic, but every 5 years NRCs, the programs that we have crop specific, like we had a program for 5 years, Canadian Wheat Alliance, that most of the tools we deployed or we learned playing with wheat. So then before that we had oilseeds, so canola, 5 years, almost 5 years we worked on canola. So now it's more like pulses, or we can say we can work on any crop species at this moment. But the core platform, that we have, we believe could be deployed, to any crops now, whether it's horticultural crops, northern agriculture, or we also do have a license for industrial hemp and cannabis. So we are working on those species as well. This is just to show you that why a single cell system with a strong reporter could give you a lot of strength in terms of functional genomics, or in terms of identifying the functional validation of the genes that you are looking in your target crops. So this slides showing the canola and wheat microspores as a single cell systems. We do work with Protoplast as a single cell system a lot. So I'll show you in my next slide. This is just to give you some examples that our team, we worked on multiple crop species I mentioned. And the traits, all our traits different, right? Depending on which crops we are working. For example, in wheat, the drought, yield improvement and drought was a focus. Currently, we are working on barley, recently funded ADF projects. Salt tolerance is the goal. During the wheat program, we worked on also durum wheat for pasta color improvement. Of course, we just finished a project that Mike Nickerson mentioned, that protein quality, improving protein quality in peas, horticultural species. Also, as I said, the berries or the industrial hemp. So those are all tissue culture amenable, depending on which genotypes we are working, we could potentially manipulate, to create variations if we don't have, variation in the existing population for the trait that you are interested. Just to walk you through, how it works, as I mentioned, Protoplast System, we developed a Protoplast as a unique tool for guide RNA validations, not necessarily that we are regenerating protoplast or we have protocol to regenerate protoplast for every species, but it's a nice tool to do the transient, assay for the reporter system and also validate the guide RNA. You can pull the protoplast and do the sequencing to show that how efficiently that the gene of your interest you could edit, with the guide RNA that you have designed. So we published, recently our experience with P protoplast. It's the same approach that we routinely use in other species, so that you design your guide RNA, you transform in your the constructs, you put a GFP as a reporter systems, and then you transfer with polyethylene glycol, pull the protoplast DNA, and then you amplify the target region around your guide RNA, and then you sequence. So that's working very, very well for us. This is just to go back that how the Microspore system first we reported in wheat could be an interesting explants or system that where you can edit the germ cell or haploid cells, where if you have a regeneration protocol, you can easily have homozygous plants. That work was, I think, earlier, about 2018, where we started to play with the gene editing, using the Cas9DS thread as a proof of concept, and then afterwards we also edited the lipoxygenase in wheat. Just one slide to show that disease resistance was a trait here. We successfully edited the gene in tomato. That's the vegetables, the greenhouse vegetables grower, they found that that's a desired trait that they want to introduce. Now I am coming to our plant protein program and the projects that Mike Nickerson, Tom Workentin, and we are working together to address the flavor issue or create variations for lipoxygenase or low lipoxygenase P lines. So again, the same, we started with the elite germplasms and the protocol that we had at NRC long time ago, and see how we can modify the protocol for that elite line. So it took a while, but we have a protocol that efficient enough to create 10, 20, transformants for Ps that you can screen for genotyping and phenotyping. This is CDC spectrum that we transform. We advanced those transgenic lines in the greenhouse condition, didn't see any significant pleiotropic effect. And then we did the sequencing to show the mutation, where the mutation is happening and how many base pairs, the deletions or insertion is happening. On the basis of that, so then we advance the seeds, because we have to now look for the traits. And our only challenge that the amount of seeds we need to do those phenotyping. So we had to develop a high throughput small scale logs enzyme assay protocol for our projects. We did germplasm screening as well to learn what is the existing level of lipoxygenase activity in the elite lines, and we use the same protocol to test the edited lines. Here I'm showing you 3 of the mutants lines with the background, the CDC spectrum, and you can see that it's significantly reduced the enzyme activity. And we also validated it with the third-party lab or commercial lab. The only problem with the assay in commercial lab is the amount of seeds we need is at least 20, 30. They prefer 30 grams. So that's a lot of seeds for us to sacrifice. Well, the preliminary data, with the minimum amount of seeds we had, we also looked at the GCMS for the volatiles. So here I'm showing the 2 of the abundant volatiles like hexanol and dimethyl sulfide, where compared to CDC spectrum, there are reduction. It's also not that significant like the lox activity, but it's showing the reduction compared to the wild type. And we also did assay for the precursors because we believe this is your fatty acid oxidation. The lipoxygenase, how the lipoxygenase could potentially contribute to the flavor is by working on the linoleic and linolenic acids. So you can see the wild type where we have the both of the the precursors, but in the 3 transgenic lines we see that there are unused linoleic and linolenic acid that could potentially explain why the lipoxygenase or the volatile profiles are changing. So currently we are increasing the seeds in the greenhouse, and we would like to have enough seeds for testing as an ingredient. So that's basically from our ADF project's conclusions and what we would like to go back to ADF and reapply for funding, because we have to take those lines to the field to see how they perform in the field. But as a output from these projects, the assay was a unique small scale assay in 96 plate that now we can screen hundreds of thousands of germplasms. We are also at the same time, so we took the approach to create the mutagenized population so that we don't know how the CRISPR edited lines will be regulated by Health Canada and CFIA. Or many of you know that last year's Health Canada from the consultations published the guidelines, but we are still awaiting from the CFIA guidelines in terms of the PNT, plans with novel traits, regulations, how gene edited lines will be regulated. So that's basically where we are now. I think this is my last slide, that just to share with you, you have seen this from the guidelines, that Health Canada provided, that according to that guideline, that foods that are derived from plants with genetic modifications but do not alter the endogenous protein in a way that it doesn't introduce or increase similarity with a known allergen or toxin, then they will not regulate this. If it doesn't increase the levels of known endogenous allergens, toxins, or anti-nutrient beyond the documented range observed, so they will always look at the existing variation in the population. And if it doesn't impact the key nutritional composition or metabolism, or intentionally change the food use of the plant, and do not result the presence of foreign DNA, if we can show that there is no agrobacterium, or we are not overexpressing or transforming with the foreign genes, so in the final product, so they will not regulate. So that's basically where I think we are now. So I'll stop here. Perhaps I went too fast, but I think we have enough time. So if you have a specific question, I'll be happy to take your questions now.
Thanks, Pankaj. Do we have any questions from the audience? Maybe I'll start just to get the ball rolling. So when we talk about, if we look, say, in the field, there's a lot of environmental effects that affects protein levels, protein profiles, and then that impacts, their performance as ingredients. So let's just say you modify the P to change the lipoxygenase activity and then alter flavors. Do you foresee the same environmental impacts in the transgenic plants in the field that you do, say, in protein levels? Like, how big of an environmental effect will it have on the flavor profiles of this?
That's a good question, Mike. From the germplasm screening, I didn't show the data. We did also, screen some of, TOMs, the materials from the GWAS population, and we have seen the variations in logs depending on which location, which year. But that's compared to our LOX mutant lines, that variation is not significant.
Okay.
That we can compare that. But the point is, until we take our materials in the field, we really cannot confirm that how in the field these lines will perform or change the LOX activity. We wanted to take it last year, but unfortunately CDC didn't have a protocol in place in terms of growing the gene edited plants, so we had to stop, and everything until now we are doing in the greenhouse. So we are hoping that going back to ADF, if we don't have the protocol within the CDC, we may have to go to a third party commercial authority who does the GMO routinely following the CFIA. But you are right, until we take these lines to the field, we cannot really guarantee that how the numbers will change. But as I said, from the regional variety trial, the populations that I receive and we screen, there are variation, but we didn't see a significant variation changes in terms of the location or in terms of the growing year.
Yeah, and just one more question. So big picture, so soya, people don't want to use soya for allergens, but there's talk about genetically modified soya and things like that. Now the pulse industry, one of the big selling feature that they market is non-GMO. So you're sort of going against what the growers and marketing of the pulse industry is pushing for. Do you think you'll see a big pushback from growers and organizations about moving towards transgenic plants?
Yeah, no, I agree with you. That's why I mentioned that unless we hear from CFIA, the guidelines, it's very difficult at this moment to say whether this two base pair deletion of the coding sequence of the lipoxygenase LOX2 or LOX3, there are two LOX, whether it will be considered as a PNT or not. If it is not considered as a, in the, I think the 2 weeks ago, the BAM, the PGDC meeting, the Prairie Grain Development Committee, where most of the variety registration or recommendation happens, there was presentation from CFIA and I think CropLife Canada, and we discussed the same. So until we receive the guidelines from CFIA, it's not clear. But the justification is, even if we know that this is the lipoxygenase 2 or lipoxygenase 3 is contributing, and we develop marker, even we can do the, as I said, we also creating the mutagenized population. We can use this data or information to screen the existing population or mutagenized population and come up with a low lipoxygenase lines. Like the companies like Genome or other companies, Benson Hill, I think they are following the same path that you have, thousands of germplasms and you screen or phenotype, high throughput phenotyping for your high protein or your flavor profile, and then you come up with few lines and then you take it to the field. So our argument will be the same, that if we learn from CFIA that it will be regulated, we just use this information, we create the marker from this research that will be used. The John Innes Center's the LOX mutant lines in that old background, Tom Orkentin is already started crossing and transferring this to elite lines, but that will take time. But I think the information we are generating will be useful for the breeding program.
Okay. Does anyone have any other questions for Venkash?
There's a question on the chat.
Oh, chat, here we go. What is yield drag? What is yield drag?
That's a good question. The conventional, the way that we do the breeding, right? You see that we can take an example. It's Sarah. Example like protein. Now the equinome is they are breeding for high protein, right? But the interesting thing is most of the high protein lines you have low yield. So where is the balance between your protein from between 25 and 30% protein? If you want to go for high protein, you will see that you have at least, your yield reduced. But so that's basically yield drag. If you want to create the genetic gain, avoiding your yield drag, so whether editing or high yielding line where you have already high yield, can you precisely just make that small change in your sequence that you will not be impacted in terms of yield, right? Although the companies like AGT or the ADM, they are arguing that they will buy those high protein lines, and they will pay premium for your low yield. So that's where, the Equinome is going towards that route, that, okay, we don't worry about the yield loss or yield drag. We will pay premium, and we will have a contract farmers who will take our seeds, grow it, and we will buy them back, and we'll pay them enough price that will cover their loss for the yield. That's another way of looking at it.
I think, Pankaj, I have one question. It's technical question in your last slide.
Yeah.
If you can share the last slide, you mentioned that the DNA is not detected in the food, and then this will be not regarded as, or I try to understand, because, yeah, if you can, because it's not beyond my knowledge, you mentioned that don't result in the presence of foreign DNA in the final plant product. So I wanted to know if you have, first the question is that if you have genetic modification, how you can end up with P products without the foreign DNA inside, or if there's no foreign DNA inside, whether this will be novel food or not novel food.
Yeah, no, that's a good question. I think that argument, or that point came from the GMO. Within the Health Canada, now they have two guidelines. One is for GMO, another one is gene-edited crops, right? So foreign DNA comes, when you transform a plant lines with agrobacterium, for an example, I'm saying. You have to show, or you have to cross it out, or you have to show that there is no agrobacterium DNA present in your P lines, that say 3 lines I showed. We have to show that. In the conventional way of creating transgenic lines, that you overexpress, right? Or you put a DNA from another species or another organism. But what we are saying, or what the recent the acceptance of FDA or in US, what we are saying, that we are not introducing any foreign DNA. It's just the plant's own DNA CDC spectrum that we edited. So we knock down 2 base pairs from its own sequence. Now, where do you do the knockdown, right? Is it in promoter regions or is it in coding sequence, right? So they will look at those, but what they're saying, we don't focus on the process. How did you create that mutation? That mutation could be in your natural variation, or you do gamma radiation, or you do EMS, chemical mutagenized, and you create that variation, and then you see the phenotype, and then you sequence your line, and you show that, okay, this is where the mutation happened. That is acceptable as a non-PNT, not novel traits. The same thing we are hoping that will happen in these gene-edited lines, or you have to find a different ways using non-DNA, and that's also possible. In other species, we have seen that you deliver your gene editing nucleus, Cas9, in your cells as a protein. So you're not using agrobacterium, or you're not using any DNA as your cargo, as your nucleus to do the cutting. So those are I think the discussion that will be considered as there is no presence of foreign DNA. But you are right. You have to show with PCR and other way, there is no selection marker that you use for plant transformation. There is no agrobacterium if you use agrobacterium. If you use biolistics, that's fine. Or if you use the other method, direct delivery, using protoplast or your Cas9 as a protein instead of DNA, that's where now most of the research is focusing, so that it will not be considered as presence of foreign DNA.
Thanks, Pankaj, for the clarification.
Thank you.
Does anyone have any other questions? Okay, if not, thank you, Panjak. We really appreciate this, and hope everyone enjoyed it. It's kind of a different talk because we don't hear from the breeding side, and you know, it really relates to, you know, what we do from the value added ingredient application side. So thanks very much. And if you have any questions, you can send me an email. I can forward that to Panjak as well. So thanks everyone, and we'll see you next time.
Thank you, Mike. I just want to add, you know, no, that's a good point, Mike, that it's not conventional way of value addition, but we believe that in addition to processing, like flavor industry, that heat treatment, the other technology we are applying, maybe if you have a genetics that's already had that trait, it enhance, you know, your ingredient qualities. That's the idea. It's not like completely alternate way of what industry is currently doing. So that's more like, you know, to put it in that way. Please do not consider that this I'm proposing is as an alternate solution of, you know, heat treatment or other processing to improve your flavor profile.
Yeah. Perfect.
Thanks,
Thanks everyone. Have a good night.
Bye.